In this video, we'll cover everything within the inheritance, variation, and evolution topic, from DNA structure and how genes are passed on, to natural selection and the evidence for evolution, including modern techniques like genetic engineering and cloning. Before we dive in, let's outline the key topics we'll discuss. First, we'll examine the key differences between sexual and asexual reproduction. Next, we'll look at how each method works and the types of offspring they produce. Then we'll explore the advantages and disadvantages of both reproductive strategies. Finally, we'll discover how some organisms can switch between both methods depending on their circumstances. Living organisms can reproduce in two fundamentally different ways. Let's first look at sexual reproduction, which involves two parents combining their genetic material. In sexual reproduction, genetic information from two organisms, a father and a mother, is combined to produce offspring that are genetically different from either parent. The male and female parents produce specialized reproductive cells called gametes. In animals, these are sperm cells from males and egg cells from females. In humans, each gamete contains 23 chromosomes, exactly half the number found in normal body cells. This reduction is vital to maintain the correct chromosome number across generations. During fertilization, the egg and sperm fuse together, forming a cell with the full number of chromosomes, half from each parent. This is why offspring inherit characteristics from both parents. The specific mixture of chromosomes they receive determines many of their traits. This blending of genetic information creates a variation among offspring. Siblings often look different because they inherit different combinations of genes from their parents. Sexual reproduction isn't limited to animals. Flowering plants also reproduce sexually. They produce male gametes in pollen and female gametes in the ovules of their flowers. When pollen reaches the female part of a flower, fertilization can occur, leading to seed formation. In contrast, asexual reproduction involves only one parent, and the offspring are genetically identical to that parent. Asexual reproduction happens through mitosis, a cell division process where one cell makes two genetically identical copies. The new cells contain exactly the same genetic information as the parent cell. Since there's only one parent, no fusion of gametes, and no mixing of chromosomes, the offspring are clones of the parent, genetically identical in every way. Many organisms reproduce asexually, including bacteria, some plants, and certain animals. For example, bacteria simply divide in two, with each new cell being a clone of the original. Now that we understand the basic differences, let's consider why these two strategies exist. Each has distinct advantages that make it beneficial in particular circumstances. Sexual reproduction offers several important advantages. First, offspring have a mixture of genes from both parents, which produces variation in the population. This variation increases the chance of a species surviving environmental changes. If conditions shift, some individuals may have characteristics that help them adapt and survive. For example, if the environment changes and introduces new predators, individuals with slightly different characteristics might be better suited to escape these predators. They're more likely to survive and reproduce, passing their beneficial genes to the next generation. This principle forms the basis for natural selection. Individuals with advantageous characteristics are more likely to survive, reproduce, and pass their genes to the next generation. Over time, this leads to better adaptation of the species to its environment and can result in evolutionary change. Humans have harnessed this process through selective breeding to develop animals and plants with desirable characteristics. For example, we've bred animals that produce more meat or milk and plants that yield larger fruits or are more resistant to disease. However, sexual reproduction also has drawbacks. It requires finding a mate, which takes time and energy. The search for a partner can also expose animals to predators. Additionally, only 50% of an individual's genes are passed to each offspring, meaning that favorable combinations of genes might be lost in the next generation. Asexual reproduction offers different advantages. Since only one parent is needed, organisms can reproduce without finding a mate, saving considerable energy. This method is typically faster than sexual reproduction, allowing organisms to produce many offspring quickly when conditions are favorable. The speed of asexual reproduction makes it particularly useful for colonizing new environments rapidly. When a single bacterial cell reaches a new location with plenty of nutrients, it can quickly multiply to form a large population. Another advantage is that 100% of the parent's genes are passed to the offspring. If the parent is well adapted to its environment, all offspring will inherit these adaptations. However, the lack of genetic variation in asexually reproducing populations can be a disadvantage. If environmental conditions change, the entire population might be susceptible to the same threats, such as new diseases or environmental stresses. Interestingly, some organisms can reproduce both sexually and asexually, depending on their circumstances. The malaria parasite provides a fascinating example. When a mosquito carrying the parasite bites a human, the parasite reproduces asexually in the human host. However, when it returns to a mosquito, it reproduces sexually. Many species of fungi reproduce both sexually and asexually. They produce spores asexually, which can become new fungi when they land in a suitable place. But they can also reproduce sexually, introducing variation into their populations. Sexually produced spores introduce variation and are often produced in response to unfavorable environmental changes. This increases the chance that the population will survive the change. Plants often demonstrate both strategies as well. Many flowering plants produce seeds through sexual reproduction, but can also reproduce asexually. Strawberry plants send out runners, stems that grow horizontally along the ground. New plants form at intervals along these runners, genetically identical to the parent. Daffodils and other bulb plants can form new bulbs from the main bulb. Each new bulb can grow into a genetically identical plant. Asexual reproduction in plants can take place in different ways. Some plants produce runners. These are stems that grow horizontally on the surface of the soil away from the parent plant. At various points along the runner, a new strawberry plant forms that is identical to the parent plant. Other examples include plants that grow from bulbs, like daffodils. New bulbs can form from the main bulb and divide off. Each new bulb can grow into a new identical plant. The ability to use both reproductive strategies gives these organisms flexibility. They can reproduce sexually to generate genetic diversity, helping the species adapt to changing conditions. In our next episode, we'll delve deeper into meiosis, the special type of cell division that produces gametes with half the normal number of chromosomes. In this episode, we'll look at why gametes need half the usual number of chromosomes. Next, we'll examine the process of meiosis and how it differs from mitosis. Then we'll explore how meiosis creates genetic variation. Finally, we'll see what happens after fertilization when gametes combine. In sexual reproduction, offspring receive genetic information from two parents. As we discussed in our previous episode, when two gametes join during fertilization, they form a cell with the full complement of chromosomes. For this to work properly, gametes must contain half the normal chromosome number. Otherwise, the fertilized cell would have too many chromosomes, which would disrupt normal development. Normal body cells in humans contain 46 chromosomes, 23 from each parent. To maintain this number across generations, gametes must have just 23 chromosomes. Meiosis is the process that produces these specialized cells with half the chromosome count. It occurs in the reproductive organs, the testes in males and the ovaries in females. Unlike mitosis, which produces two identical cells, meiosis involves two sequential cell divisions, resulting in four genetically different cells. Let's look at how this works. Before meiosis begins, the cell duplicates its genetic information. Each chromosome makes an exact copy of itself, forming two identical strands attached at a central point. In the first division of meiosis, something unique happens. The duplicated chromosomes arrange themselves in pairs in the center of the cell. These pairs consist of matching chromosomes, one originally from the mother and one from the father. They line up along the middle of the cell. The pairs are then pulled apart, with one copy of each chromosome moving to opposite ends of the cell. After this first division, we have two cells, each containing half the number of chromosome pairs, but each chromosome still consists of two attached strands. In the second division of meiosis, these cells divide again, the chromosomes line up in the center once more, and the two strands of each chromosome are pulled apart. This results in four cells, each containing just one set of chromosomes, half the number found in normal body cells. These are our gametes. An important feature of meiosis is that it creates genetic diversity. During the process, genetic material from the mother's and father's chromosomes can exchange in a process called crossing over. Additionally, the way chromosomes separate during meiosis is random. This shuffling of genetic material means that each gamete has a unique combination of genes. This is why siblings can look quite different from each other. They inherit different combinations of genes from their parents. After fertilization, when two gametes fuse, the resulting cell contains the normal number of chromosomes again, half from each parent. This cell, called a zygote, then divides by mitosis to produce lots of new cells as an embryo develops. As the embryo grows, these cells differentiate to form the various specialized cell types that make up a complete organism. Meiosis is crucial for sexual reproduction and genetic diversity. Without it, the chromosome number would double with each generation, which would be incompatible with life. And without the genetic reshuffling that occurs during meiosis, offspring would not have the genetic variation that helps species adapt and evolve. In our next episode, we'll explore DNA and the genome, the genetic instructions that determine the characteristics of living organisms. In this episode, we'll explore DNA and the genome, the remarkable molecules that contain the genetic instructions for all living organisms. We'll examine the structure of DNA, take a look at genes, explore protein synthesis, and finally, we'll discuss mutations. Understanding genetics starts with getting to grips with DNA and genes. DNA stands for deoxyribonucleic acid and is the chemical that all of the genetic material in a cell is made up from. DNA contains genetic information, basically all the instructions to put an organism together and make it work. It's what's in your DNA that determines what inherited characteristics you have. DNA is found in the nucleus of animal and plant cells in really long structures called chromosomes. Chromosomes normally come in pairs. In humans, we have 23 pairs, making a total of 46 chromosomes. DNA is made up of two strands coiled together in the shape of a double helix. This twisted ladder structure is critical to how DNA functions. Let's look more closely at what DNA is made of. DNA strands are polymers made up of lots of repeating units called nucleotides. Each nucleotide consists of three parts: a sugar, a phosphate group, and a base. The sugar and phosphate groups in the nucleotides form the backbone of the DNA strands. The sugar and phosphate groups alternate, with the phosphate groups linking the sugars together. one of four different bases, A, T, C, or G, joins to each sugar. These letters stand for adenine, thymine, cytosine, and and the two strands of DNA are held together by bonds between the bases. Importantly, the bases don't pair up randomly. A always pairs up with T, and C always pairs up with G. This is called complementary base pairing. This complementary nature of DNA is crucial for both DNA replication and protein synthesis. It ensures that genetic information can be copied accurately. A gene is a small section of DNA found on a chromosome. Each gene codes for a particular sequence of amino acids, which are put together to make a specific protein. Only 20 amino acids are used, but they make up thousands of different proteins. Genes simply tell cells in what order to put the amino acids together. Each amino acid is coded for by a sequence of three bases in the gene. This three-base sequence is often called a codon. The amino acids are joined together to make various proteins depending on the order of the gene's bases. This in turn determines what type of cell it is, like a red blood cell. It's the order of bases in a gene that decides the order of amino acids in a protein. Let's turn our attention to how proteins are made using the information in DNA. This process is called protein synthesis. Proteins are made in the cell cytoplasm on tiny structures called ribosomes. To make proteins, ribosomes use the code in the DNA. But there's a problem. DNA is found in the cell nucleus and can't move out of it because it's really big. So the cell needs to get the code from the DNA to the ribosomes. This is done using a molecule called mRNA, which is made by copying the code from the nucleus. The mRNA acts as a messenger between the genetic code in the nucleus and the ribosomes. It carries the message between the two. The correct amino acids are brought to the ribosomes in the correct order by carrier molecules. Once the chain of amino acids has been assembled, it folds into a unique shape. This folding is crucial. It gives the protein its specific three-dimensional structure, which allows it to perform its particular function. The shape of a protein is directly related to its role in the body. Proteins have many different functions in the body. Let's look at some examples. Enzymes act as biological catalysts to speed up chemical reactions in the body. For instance, digestive enzymes help break down food. Hormones, like insulin, are used to carry messages around the body. Insulin is released into the blood by the pancreas to regulate blood sugar levels. Structural proteins, like collagen, are physically strong. They strengthen connective tissue, such as ligaments and cartilage. Every organism has a genome. The genome is just a fancy term for the entire set of genetic material in an organism. Scientists have worked out the complete human genome. Understanding the human genome is a really important tool for science and medicine for many reasons. It allows scientists to identify genes in the genome that are linked to different types of disease. Knowing which genes are linked to inherited diseases could help us understand them better and develop effective treatments for them. Scientists can also look at genomes to trace the migration of certain populations of people around the world. All modern humans are descended from a common ancestor in Africa, but humans can now be found all over the planet. The human genome is mostly identical in all individuals, but as different populations of people have split off in different directions, they gradually developed small differences in their genomes. By investigating these differences, scientists can work out when new populations split off and what paths they took. Sometimes the sequence of DNA bases can be changed. These changes are called mutations. Mutations are random changes in an organism's DNA. and they can sometimes be inherited. Mutations occur continuously. They can occur spontaneously, such as when a chromosome isn't replicated properly. However, the chance of mutations is increased by exposure to certain substances or some types of radiation. Mutations change the sequence of the DNA bases in a gene, which produces a genetic variant, a different form of the gene. As the sequence of DNA bases codes for the sequence of amino acids in a protein, mutations to a gene can sometimes lead to changes in the protein that it codes for. Most mutations have little or no effect on the protein. Some will change it in such a small way that its function or appearance is unaffected. However, some mutations can seriously affect a protein. Sometimes the mutation will code for an altered protein with a change in its shape. This could affect its ability to perform its function. For example, if the shape of an enzyme's active site is changed, the substrate may no longer be able to fit into it. Structural proteins like collagen could lose their strength if their shape is changed, making them pretty useless at providing structure and support. If there's a mutation in the non-coding DNA, it can alter how genes are expressed. There are different types of mutations. Insertions are where a new base is inserted into the DNA base sequence where it shouldn't be. You should remember that free bases in a DNA base sequence codes for a particular amino acid. An insertion changes the way the groups of free bases are read, which can change the amino acids that they code for. Insertions can change more than one amino acid, as they have a knock-on effect on the bases further on in the sequence. Deletions are when a random base is deleted from the DNA base sequence. Like insertions, they change the way that the base sequences are read and can have knock-on effects further down the sequence. Substitutions happen when a random base in the DNA base sequence is changed to a different base. A substitution may change the amino acid that is coded for, potentially affecting the protein structure and function. Understanding DNA, genes, the genome, and mutations helps us appreciate the remarkable complexity of life at the molecular level. These tiny structures and processes determine who we are and how our bodies function. They represent the basic code of life itself, instructions passed down through countless generations. In our next episode, we'll explore how these genetic principles apply to genetic inheritance. In this episode, we'll explore genetic inheritance. Key topics we'll discuss today include how genes control characteristics, different forms of genes called alleles, genetic diagrams, and family trees. Many genes can directly control what characteristics you develop. Different genes control different characteristics. Some characteristics are controlled by a single gene, like mouse fur color and red-green color blindness in humans. However, most characteristics are controlled by several genes interacting together. This makes inheritance patterns more complex for traits like height and skin color. All genes exist in different versions called alleles. These are represented by letters in genetic diagrams, one letter for each allele. You have two versions or alleles of every gene in your body, one from each parent. These alleles can be the same or different from each other. If an organism has two alleles for a particular gene that are the same, then it's homozygous for that trait. If the alleles for a particular gene are different, then it's heterozygous. Sometimes one allele can completely override the other when they're different. The allele that shows itself is called the dominant allele. We typically use a capital letter for dominant alleles. For example, capital A. The other one is called the recessive allele, represented by a small letter, for example, lowercase a. A recessive allele only shows itself when there's no dominant allele present. For an organism to display a recessive characteristic, both its alleles must be recessive. To display a dominant characteristic, the organism only needs one dominant allele. This means the dominant allele overrides the recessive one if the organism is heterozygous. Your genotype is the combination of alleles you have, while your phenotype are the observable characteristics you have. Let's see how we can predict inheritance patterns using genetic diagrams, also known as Punnett squares. Suppose we're breeding chickens with long legs. The allele which causes chickens to have long legs is recessive and represented by a lowercase h, while normal legs are due to a dominant allele, represented here with a capital H. Let's look at what happens if you breed from two heterozygous chickens. We start by identifying the parents' genotypes. Both parents are heterozygous. Next, we determine what types of sex cells each parent can produce. We can then create a Punnett square to show all possible combinations. We place one parent's potential gametes along the top and the other parent's along the side. Filling in the square gives us the following. Looking at the phenotypes, we can see that three of the offspring will have normal legs, while one will have long legs. This gives us a 3-to-1 ratio of normal to long-legged offspring in this generation. Now, if two of these offspring breed together, what happens in the next generation? It depends on which offspring breed together. If a homozygous dominant breeds with a heterozygous chicken, all their offspring will be normal. On the other hand, if a heterozygous chicken breeds with a homozygous recessive chicken, half their offspring will be normal and half will have long legs. Let's look at another example. A lion with long hair, which is the dominant trait, is bred with a lion with short hair, which relates to the recessive trait. The long-haired lion has a heterozygous genotype, while the short-haired lion has a homozygous recessive genotype. When we work out the Punnett square, we find the offspring will have a 1-to-1 ratio, half with long hair and half with short hair. This happens because the heterozygous parent can pass either the dominant or recessive allele to its offspring. The homozygous recessive parent can only pass the recessive allele. Understanding genetic inheritance can help us interpret family trees, which is particularly useful for tracking genetic disorders like cystic fibrosis. From a family tree, we can often tell if an allele for a particular disorder is dominant or recessive. For cystic fibrosis, we know it's a recessive disorder because plenty of people carry the allele but don't have the disease. By analyzing a family tree, we can also determine the probability that someone will inherit a genetic disorder. For example, if both parents are carriers for a recessive disorder but don't show symptoms, there's a 25% chance their child will have the disorder. Similarly, there's a 50% chance their child will be a carrier like them, and a 25% chance the child will neither have the disorder nor be a carrier. The risk calculation for the new baby is just the same as in the genetic diagram, so a new baby could be unaffected, be a carrier, or have cystic fibrosis. Genetic diagrams are important tools in understanding and predicting inheritance patterns. They allow us to calculate the probability of particular traits appearing in offspring based on the parents' genotypes. These diagrams help us understand why certain traits might skip generations or appear unexpectedly. For example, if two brown-eyed parents have a blue-eyed child, genetic diagrams explain that both parents must carry a recessive allele for blue eyes. It's important to remember that genetic diagrams show probabilities. They don't guarantee exactly what will happen with each individual offspring. Just like flipping a coin doesn't guarantee exactly 50% heads and 50% tails, in a small number of flips, genetic inheritance can sometimes produce unexpected ratios in small family sizes. In reality, many traits are influenced by multiple genes and environmental factors, making inheritance patterns more complex than the simple examples we've discussed. However, these principles form the foundation for understanding how genes are passed from one generation to the next. In our next episode, we'll explore how these inheritance patterns can lead to genetic disorders when certain mutations are passed down through families. In this episode, we'll explore inherited disorders. We'll start by outlining what inherited disorders are, then discuss examples such as cystic fibrosis and polydactyly, and finally look at embryo screening and the ethical questions it raises. Some disorders can be inherited from your parents through the genes they pass on to you. These genetic conditions are caused by mutations in specific genes that lead to health problems. Let's start by looking at cystic fibrosis, which is a genetic disorder affecting cell membranes. It results in the body producing a lot of thick, sticky mucus in the air passages and in the pancreas. Cystic fibrosis is caused by a recessive allele. This means that for a person to have the condition, they must inherit two copies of the recessive allele, one from each parent. Because it's recessive, people with only one copy of the allele won't have the disorder. They're known as carriers. For a child to have cystic fibrosis, both parents must be either carriers or have the disorder themselves. As genetic diagrams show, if both parents are carriers, there's a 1 in 4 chance that their child will have the condition. Now let's look at polydactyly, which is quite different in terms of how it's inherited. Polydactyly is a genetic disorder where a baby is born with extra fingers or toes. It doesn't usually cause any other problems, so it's not life-threatening. Unlike cystic fibrosis, polydactyly is caused by a dominant allele. This means the disorder can be inherited if just one parent carries the defective allele. As the genetic diagram shows, there's a 50% chance of a child having the disorder if one parent has it. Modern medicine now allows for screening of embryos for genetic disorders. This process is done during in vitro fertilization. In IVF, embryos are fertilized in a laboratory and then implanted into the mother's womb. Before being implanted, it's possible to remove a cell from each embryo and analyze its genes. Many genetic disorders can be detected in this way, such as cystic fibrosis. It's also possible to get DNA from an embryo in the womb and test it for disorders. There are lots of ethical, social, and economic concerns surrounding embryo screening. Embryonic screening is quite controversial because of the decisions it can lead to. For embryos produced by IVF after screening, embryos with bad alleles would be destroyed. For embryos in the womb, screening could lead to the decision to terminate the pregnancy. There are strong arguments both for and against embryonic screening. Those against embryonic screening worry that it implies people with genetic problems are undesirable. This could increase prejudice. They also express concerns about where to draw the line. Could parents eventually select for traits like eye color or intelligence? Others note that screening is expensive and question whether it's a good use of resources. On the other hand, supporters of embryonic screening argue that it will help to stop people suffering. They point out that treating genetic disorders costs the government and taxpayers a lot of money. Some also believe there are limits to how far screening should go. But at the moment, parents cannot even select the sex of their baby unless it relates to a health reason. Understanding inherited disorders and the technologies available to detect them raises important questions about the intersection of science, medicine, and ethics. While advances in genetic screening offer new possibilities for preventing suffering, they also challenge us to consider difficult questions about what kinds of interventions are appropriate and where the boundaries should be. In our next episode, we'll explore sex determination and how the combination of chromosomes determines whether a baby will be male or female. In this brief episode, we'll explore how your biological sex is determined at the moment of conception by specific chromosomes. Understanding this process helps explain one of the fundamental aspects of human development. Let's begin by recalling what we've learned about chromosomes. There are 23 pairs of chromosomes in every human body cell. Of these, 22 pairs are matched pairs called autosomes that control various characteristics. The 23rd pair is special. They're the sex chromosomes labeled X or Y. These two chromosomes decide your sex, whether you develop as male or female. Males have an X and a Y chromosome, giving them the combination XY. It's the Y chromosome that causes male characteristics to develop. Without it, male features wouldn't appear. Females have two X chromosomes, giving them the combination XX. This XX combination allows female characteristics to develop. During gamete formation, something important happens. When making sperm cells, the X and Y chromosomes are separated during meiosis, the special cell division we discussed in a previous episode. This separation means that each sperm cell gets either an X chromosome or a Y chromosome, never both. There's a 50% chance a sperm cell will contain an X chromosome and a 50% chance it will contain a Y chromosome. A similar process happens when females produce X cells. However, since females only have X chromosomes, all X cells will contain an X chromosome. When fertilization occurs, the sex of the future child depends on which sperm cell fertilizes the egg. If a sperm cell carrying an X chromosome fertilizes the egg, the resulting combination will be XX, a female. If a sperm cell carrying a Y chromosome fertilizes the egg, the resulting combination will be XY, a male. We can use genetic diagrams to illustrate this process by creating a simple Punnett square with the mother's possible gametes along one side and the father's possible gametes. Along the top, we can see all possible combinations. The resulting square shows two XX outcomes and two XY outcomes, demonstrating that there's an equal probability, 50-50, of having a boy or girl in each pregnancy. This 50-50 ratio is only a probability in each individual pregnancy. Having a boy doesn't make it more likely the next child will be a girl. Each pregnancy is an independent event with the same 50% probability. Understanding sex determination helps us appreciate how fundamental biological characteristics are established at conception through the combination of chromosomes. No two living things are exactly alike. In this episode, we'll explore variation, the differences that make each organism unique. We'll look at how genes contribute to differences in organisms, how the environment influences characteristics, and how mutations introduce new variation. You've probably noticed that all people are different from one another. These differences aren't just limited to humans. All species show variation. Different species obviously look different. My dog definitely doesn't look like a dairy cow. But even organisms of the same species will usually look at least slightly different. In a room full of people, you'll see different eye colors, differently shaped noses, and various hair colors. These differences are called variation within a species, and there are two main types: genetic variation and environmental variation. Genetic variation comes from our genes. All plants and animals have characteristics that are similar in some ways to their parents. This happens because an organism's characteristics are determined by the genes they inherit. Genes are the codes inside your cells that control how you're made. These genes are passed on in sex cells or gametes, from which the offspring develop. Most animals and quite a lot of plants get some genes from the mother and some from the father. The combining of genes from two parents causes genetic variation. No two of the species are genetically identical except for identical twins. Some characteristics are determined only by genes. In humans, these include eye color, blood group, and certain inherited disorders like hemophilia or cystic fibrosis. However, characteristics are also influenced by the environment. The environment, including the conditions that organisms live and grow in, causes differences between members of the same species. For example, a plant grown in plenty of sunlight would be green, but the same plant grown in darkness would be tall and spindly with yellow leaves. These are environmental variations. Environmental variation covers a wide range of differences, from losing a finger in an accident to getting a suntan to plants having yellow leaves. Most characteristics are actually due to a mixture of genes and the environment. For example, your maximum height that you could possibly grow to is determined by your genes, but whether you actually reach that height depends on environmental factors like nutrition. Mutations also introduce variation. Mutations are changes to the sequence of bases in DNA. As we learned earlier, mutations can lead to changes in the protein that a gene codes for. Most mutations have no effect on the protein and therefore don't alter characteristics significantly. Although it's very rare, mutations can result in a new phenotype being seen in a species. If the environment changes and the new phenotype makes an individual more suited to the new environment, it can become common throughout the species relatively quickly by natural selection. In our next episode, we'll explore natural selection and how it drives evolution. The theory of evolution explains how all species on Earth today descended from simple life forms that first appeared over 3 billion years ago. We'll cover Darwin's revolutionary insights into natural selection, the process of speciation, evidence supporting evolutionary theory, and finally, extinction factors. Let's start with Darwin's groundbreaking observations and conclusions. Charles Darwin developed his theory based on careful observations made during his 5-year voyage aboard HMS Beagle. Darwin recognized two crucial facts about living organisms that became foundational to his theory. First, individuals within any species show lots of variation in their characteristics, from size and color to behavior and physiology. Second, organisms constantly compete for limited resources in their environment. From these observations, Darwin reasoned that individuals with characteristics better suited to their environment would succeed more in this competition. These better adapted organisms would survive longer, find more food, avoid predators more effectively, and ultimately produce more offspring. This principle became known as survival of the fittest, though fittest means best adapted to the environment, not necessarily strongest. The organisms with advantageous traits would pass these beneficial characteristics to their offspring through inheritance. Conversely, organisms with less favorable characteristics would struggle to survive and reproduce. Their genes would gradually become less common in the population, potentially disappearing entirely. Over many generations, beneficial characteristics become increasingly prevalent whilst disadvantageous ones diminish. This gradual change in populations over time is evolution, driven by the mechanism of natural selection. Evolution explains both the unity and diversity of life on Earth. All species share common ancestors if we trace back far enough, explaining fundamental similarities like cellular structure and DNA. The differences arise because populations have adapted to vastly different environments over millions of years. Darwin's ideas have been refined with modern scientific discoveries, particularly in genetics. We now understand that genes control inheritance, and variation arises from mutations, random changes in DNA sequences. When beneficial mutations occur, natural selection favors these variants, potentially spreading them throughout the population. This genetic understanding, unavailable to Darwin, has strongly validated his fundamental insights about evolution's mechanisms. Over extended periods, accumulated changes can become so significant that populations can no longer interbreed successfully. This process, called speciation, creates entirely new species from existing ones. For speciation to occur, it typically requires reproductive isolation between populations. Reproductive isolation often occurs when populations become geographically separated by barriers like mountain ranges, rivers, or ocean expanses. These separated populations face different environmental pressures and food sources, driving divergent evolutionary paths. As isolated populations adapt to their specific environments, they accumulate different genetic changes. Eventually, they become so genetically distinct that they cannot produce fertile offspring together, even if reunited. Once reproductive isolation is complete, the populations represent separate species following independent evolutionary trajectories. This process has generated Earth's millions of species from common ancestors over billions of years. Multiple lines of evidence support evolution by natural selection, though Darwin initially faced skepticism. When On the Origin of Species was published in 1859, the supporting evidence was less comprehensive than today. The fossil record provides compelling evidence for evolution. Fossils preserve remains of ancient organisms in rock layers, with older fossils in deeper layers. This record shows countless extinct species, demonstrating that Earth's inhabitants have changed dramatically over time. Fossils reveal gradual transitions between ancient and modern forms. The evolution of horses, for example, can be traced from small, multi-toed ancestors to modern single-hoofed species through numerous intermediate fossils. Comparing anatomy, which means structure, gives us another line of evidence through homologous or similar structures. The front limbs of humans, cats, whales, and bats contain the same basic bone arrangements despite serving different functions. These structural similarities suggest descent from a common ancestor. Vestigial structures provide further anatomical evidence for evolution. The human appendix, wisdom teeth, and tailbone serve little or no function but make sense as remnants from ancestors who used them. Some snakes retain tiny, internal leg bones despite having no legs. These vestigial structures would be inexplicable without evolution but perfectly logical as inheritance from legged ancestors. Modern molecular biology has provided powerful new evidence through DNA and protein comparisons. Species sharing recent common ancestors have more genetic similarities than those whose lineages deviated earlier. Humans and chimpanzees share approximately 98% of their DNA, reflecting our recent evolutionary divergence. In contrast, humans share less genetic similarity with more distantly related species. We can observe evolution occurring in real time with rapidly reproducing organisms. Bacterial evolution of antibiotic resistance demonstrates natural selection within human timescales. When exposed to antibiotics, susceptible bacteria die, whilst resistant variants survive and multiply, creating resistant populations. Darwin's theory initially faced opposition for several reasons beyond scientific skepticism. It contradicted prevailing religious beliefs about divine creation, offering a naturalistic explanation for life's diversity. Darwin couldn't fully explain how traits were inherited or how new variations arose. The science of genetics, which would later support his theory, remained undiscovered for decades after his publication. Limited fossil evidence at the time made Darwin's claims difficult to verify conclusively. Alternative evolutionary theories competed with Darwin's natural selection. Jean-Baptiste Lamarck proposed that organisms could pass acquired characteristics to offspring, suggesting that traits developed during life would be inherited. Lamarck's theory predicted that a giraffe stretching to reach high leaves would develop a longer neck, passing this trait to offspring. Similarly, a blacksmith's children would inherit his muscular arms developed through work. However, experiments consistently disproved Lamarckian inheritance. We now know that acquired physical changes don't alter genetic information. If you lose a finger in an accident, your children aren't born with missing fingers. The discovery of genetics validated Darwin's theory by explaining inheritance mechanisms. Mendel's work on heredity, though initially overlooked, eventually merged with evolutionary theory to form modern evolutionary synthesis. Additional fossil discoveries have revealed many transitional forms between major groups. Archaeopteryx, discovered shortly after Darwin published his theory, showed features of both reptiles and birds. Tiktaalik, found in 2004, demonstrates the transition from fish to tetrapods with both fins and primitive limb bones. Extinction represents evolution's other phase, the disappearance of species unable to adapt sufficiently. The fossil record reveals that over 99% of all species that ever lived are now extinct. Species become extinct when environmental changes exceed their ability to adapt. For example, climate shifts, habitat destruction, or new predators can eliminate populations before beneficial mutations arise and spread. New diseases can devastate species lacking immunity. Competition from better adapted species can drive others to extinction through resource depletion. Invasive species often outcompete native organisms, leading to local or global extinctions. Catastrophic events cause mass extinctions, eliminating numerous species simultaneously. The asteroid impact 66 million years ago killed the dinosaurs along with approximately 75% of all species. Earth has experienced five major mass extinctions, each followed by evolutionary radiations as survivors diversified. These events have profoundly shaped life's history, creating opportunities for new groups to dominate. Human activities are currently driving what scientists call the sixth mass extinction. Habitat destruction, pollution, overharvesting, and climate change are eliminating species faster than natural background rates. Current extinction rates exceed normal levels by 100 to 1,000 times. Understanding evolution helps us appreciate life's interconnectedness and predict species' responses to environmental changes. This knowledge proves crucial for conservation efforts and managing human impacts on biodiversity. Evolution by natural selection, supported by evidence from anatomy, molecular biology, and direct observation, stands as one of science's most robust theories. It explains life's diversity whilst revealing the common threads connecting all organisms. Next time, we'll explore how humans apply evolutionary principles through selective breeding of plants and animals. Today, we'll explore selective breeding, a technique humans have used for thousands of years to develop plants and animals with characteristics we find useful or attractive. In this episode, we'll take a look at what selective breeding actually is, the steps involved in this process, examples of selective breeding in action, and issues associated with selective breeding. Selective breeding might sound like a complicated topic, but it's actually really straightforward. It's when humans choose plants or animals with characteristics they want to keep and make sure these organisms reproduce together. This way, the genes for those useful or attractive features remain in the population. Think about it. We've developed cows that produce tons of milk, crops that resist plant diseases, and dogs with friendly personalities. None of these existed naturally. Humans created them through selective breeding. The process is super simple. First, from your existing stock of plants or animals, you pick out the ones with the characteristics you're looking for. Then you breed these selected organisms with each other. From their offspring, you again select the ones showing the best examples of the trait you want and breed them together. You keep this up for several generations, and the desirable trait gets stronger and stronger. Eventually, all the offspring have the characteristic you wanted. Farmers use selective breeding all the time to improve yields. For example, to get more milk, a farmer might breed together the cows and bulls with the best milk producing traits. After doing this for several generations, they end up with cows that produce way more milk than their ancestors ever did. To get more meat, they might select cattle with genes for larger size. Selective breeding isn't some new technology. People have been doing it for thousands of years. It's how we ended up with edible crops from wild plants, and how we got domesticated animals like cows and dogs. Before modern science, farmers and animal breeders didn't know about genes, but they still observed which individuals had the best traits and bred them together. But selective breeding isn't perfect. It has some serious drawbacks. The main problem is that it reduces what scientists call the gene pool, the number of different alleles or forms of a gene in a population. We covered alleles in our episode on genetic inheritance. This happens because the breeder keeps breeding from similar animals or plants which are all closely related. This is known as inbreeding. Inbreeding can cause health problems because there's more chance of harmful recessive defects when the gene pool is limited. Some dog breeds are particularly susceptible to certain defects because of inbreeding, like bulldogs having breathing problems. There can also be serious issues if a new disease appears. Because there's not much variation in the population and all the stock are closely related to each other, if one of them is going to be killed by a new disease, the others are likely to succumb to it too. This lack of genetic diversity means if environmental conditions change, these selectively bred organisms might not have the genetic variation needed to adapt and survive. Understanding selective breeding helps us appreciate how humans have shaped the domesticated plants and animals we rely on today. It also highlights the importance of maintaining genetic diversity in our food sources. In our next episode, we'll explore genetic engineering, a more recent technology that allows scientists to modify the genetic material of organisms directly. Genetic engineering represents one of the most powerful technologies in modern biology, enabling scientists to transfer genes between completely unrelated organisms. In this video, we'll look at what genetic engineering is, how scientists transfer genes between organisms, real-world applications, and benefits and concerns of genetic engineering. Let's start by defining genetic engineering and its unique capabilities. Genetic engineering involves extracting genes responsible for desirable characteristics from one organism and inserting them into another organism's genome. This process transfers specific traits directly between species that could never naturally interbreed. Unlike selective breeding, which requires many generations of controlled reproduction to enhance existing traits, genetic engineering achieves precise modifications immediately. Scientists can add characteristics from bacteria to plants or from humans to bacteria, combinations impossible through conventional breeding. The genetic engineering process follows a systematic sequence of carefully controlled steps. First, scientists identify and isolate the specific gene responsible for the desired characteristic using restriction enzymes. Restriction enzymes function as molecular scissors, cutting DNA at precise locations to extract the target gene. These enzymes recognize specific DNA sequences, ensuring accurate removal of the desired genetic material. The isolated gene must then be inserted into a vector, a biological vehicle for gene delivery. Common vectors include bacterial plasmids, which are small circular DNA molecules, or modified viruses that can penetrate cells efficiently. Different organisms require different vectors for successful gene transfer. Plant cells might receive genes through bacterial vectors, whilst animal cells often require viral vectors designed to integrate with mammalian DNA. The vector carries the gene into target cells like a molecular delivery system. Once inside target cells, the new gene integrates into the organism's genome, becoming part of its genetic blueprint. If modification occurs early in development, such as in embryos, the entire organism develops with a new characteristic. Successful integration means the organism's cells produce proteins coded by the inserted gene. These proteins create the desired characteristics, whether insulin production in bacteria or pest resistance in crops. Genetic engineering has produced remarkable medical breakthroughs, particularly in pharmaceutical production. Bacteria modified with human insulin genes now produce vast quantities of this essential hormone for diabetics. This bacterial insulin is identical to human insulin, eliminating previous reliance on animal sources. Before genetic engineering, diabetics depended on insulin extracted from pig pancreases, which sometimes caused allergic reactions. Now, genetically modified bacteria produce pure human insulin safely and efficiently in controlled fermentation tanks. Scientists have engineered bacteria to manufacture numerous therapeutic proteins, including human growth hormone for treating dwarfism and clotting factors for hemophilia patients. These medicines would be impossible to produce affordably without genetic modification. Agricultural applications have created crops with enhanced characteristics benefiting both farmers and consumers. Genetically modified plants can resist diseases, tolerate herbicides, and repel insect pests without chemical pesticides. Some corn varieties produce bacterial proteins toxic to caterpillars but harmless to humans. This built-in pest resistance reduces crop losses and decreases pesticide use, potentially benefiting the environment. Gene therapy represents genetic engineering's most ambitious medical application. Researchers are developing treatments that insert functional genes into patients with genetic disorders. This approach could potentially cure conditions like cystic fibrosis by replacing faulty genes with working versions. Despite these benefits, genetic engineering generates significant controversy and legitimate concerns. Critics worry about unforeseen long term consequences that might only emerge after widespread adoption. Environmental concerns focus on genetically modified crops potentially affecting ecosystems. Modified plants might crossbreed with wild relatives, spreading engineered traits unpredictably through natural populations. Reduced biodiversity represents another ecological concern. GM crops might harm beneficial insects or reduce wildflower populations on farmland. These changes could cascade through food webs, affecting birds and other wildlife dependent on agricultural ecosystems. Consumer concerns about GM foods persist despite extensive safety testing, showing no increased health risks. Many people prefer avoiding genetically modified products, driving demand for labeling and non GM alternatives. Ethical questions arise about humanity's right to alter genetic codes fundamentally. Should we modify organisms for our benefit? And where should we draw boundaries on this powerful technology? Next time, we'll explore cloning, another biotechnology raising scientific and ethical questions. In this episode, we'll cover plant cloning methods, animal cloning techniques, Dolly the sheep, and the challenges and concerns associated with cloning. Let's start with the world of plant cloning. Plants can be cloned through tissue culture, a laboratory technique using small samples of plant cells. These cells are placed in growth medium containing specific nutrients and hormones that stimulate development into complete plants. Tissue culture offers several advantages over traditional propagation methods. Plants can be produced rapidly in minimal space, grown throughout the year regardless of season, and multiplied from tiny amounts of parent material. One of the benefits of cloning is that scientists can use tissue culture to preserve endangered plant species that struggle to reproduce naturally. Commercial plant breeders employ tissue culture to mass produce desirable varieties quickly. A single exceptional plant can generate thousands of identical offspring within months, ensuring consistent quality for agriculture and horticulture. Taking cuttings represents a much older cloning method that gardeners have practiced for centuries. This involves removing a portion of a healthy parent plant, typically a stem or leaf, and encouraging it to develop roots. Cuttings provide a quick, inexpensive way to produce genetically identical plants. Many common garden plants, from roses to tomatoes, can be successfully propagated this way without specialized equipment. Both methods produce clones, organisms genetically identical to their parent. This ensures desirable traits like disease resistance, fruit quality, or flower color are preserved exactly in offspring. Animal cloning requires far more complex procedures than plant cloning. Embryo transplants allow farmers to produce multiple offspring from their highest quality livestock. This technique begins with collecting sperm from a prize bull and eggs from an elite cow. The eggs are fertilized artificially in laboratory conditions, creating embryos with superior genetics. Each embryo is then split multiple times whilst cells remain unspecialized, creating numerous identical embryos. These cloned embryos are implanted into surrogate mothers who carry them to term. The resulting calves are genetically identical to each other, though not to the surrogate mothers who birthed them. This technique can produce hundreds of elite offspring annually from a single genetic pairing. Adult cell cloning represents a more revolutionary approach, creating copies from mature organisms. This process begins by removing the nucleus from an unfertilized egg cell, essentially creating an empty cellular vessel. Scientists then extract the nucleus from an adult body cell, such as a skin or udder cell. This nucleus, containing the complete genetic information, is inserted into the enucleated egg cell. An electric shock stimulates the reconstructed egg, triggering cell division as if fertilization had occurred. Once the embryo develops into a ball of cells, it's implanted into a surrogate mother's womb. This technique famously produced Dolly the sheep in 1996. Dolly demonstrated that specialized adult cells could be manipulated to create entire organisms. Her birth revolutionized our understanding of cellular development and opened new possibilities for biotechnology. Despite technological advances, cloning presents significant challenges and concerns. The primary issue involves reduced genetic diversity in cloned populations. Genetic uniformity makes populations vulnerable to diseases and environmental changes. If all individuals share identical genes, a single pathogen could potentially eliminate entire populations lacking resistance. Cloned animals often experience serious health problems. Dolly developed arthritis at an unusually young age, raising questions about premature aging in clones. Many cloned animals suffer organ dysfunction, immune system abnormalities, and shortened lifespans. The success rate for animal cloning remains extremely low, with numerous failures for each success. These failures raise ethical concerns about animal welfare and the justification for continuing such research. However, cloning research offers potential benefits, including preserving endangered species and advancing medical understanding. Studying cloned animals provides insights into genetic disorders, aging processes, and developmental biology. Next time, we'll explore speciation, examining how species change over time. In this episode of our series, we'll look at what speciation actually means, population isolation, the role of natural selection in all of this, and the person behind the theory, Alfred Russel Wallace. Evolution, as we learned in a previous episode, leads to the development of lots of different species over time. This process of creating new species is called speciation. Let's take a look at what this means. A species is a group of similar organisms that can reproduce to give fertile offspring, while speciation is the development of a new species. Speciation occurs when populations of the same species become so different that they can no longer successfully interbreed to produce fertile offspring. In other words, they've evolved into separate species. How does this happen? It starts with isolation, where populations of a species become separated, often due to a physical barrier appearing between them. For example, floods and earthquakes can create barriers that geographically isolate some individuals from the main population. Mountains rising up, rivers changing course, or even roads in cities can separate populations. Imagine a population of beetles living in a forest that gets split by a new motorway being built right through the middle. The beetles on one side can no longer mate with the beetles on the other side because they simply can't cross the busy road. This physical separation is the first crucial step towards speciation. When populations are isolated, the conditions on either side of the barrier will be slightly different. They might have different climates, different predators, or different food sources. This may mean different characteristics will become more common in each population due to natural selection operating differently. Let's continue with our beetle example to see how this works. Perhaps one side of the motorway has darker soil, whilst the other side has lighter, sandy ground. On the dark soil side, beetles with darker coloration will be better camouflaged from predators like birds. Meanwhile, on the sandy side, lighter colored beetles will have the advantage because they blend in better with their surroundings. Each population shows genetic variation because they have a wide range of alleles. In each environment, individuals with characteristics that make them better adapted to their surroundings have a better chance of survival and are more likely to breed successfully. So the alleles that control these beneficial characteristics are more likely to be passed on to the next generation. Over time, the gene pool of each population changes in different ways. Think of it like two separate breeding programs happening simultaneously, each selecting for different traits. After many generations, our beetle populations might become so genetically different that even if the motorway was removed, they could no longer produce fertile offspring together. The dark beetles and light beetles would have become entirely separate species through the process of speciation. An important scientist in developing the theory of speciation was Alfred Russel Wallace. He was working at the same time as Charles Darwin and was one of the early scientists exploring the idea of speciation. Wallace's contributions to evolutionary theory were absolutely groundbreaking, yet he often gets overshadowed by Darwin's fame. During his career, Wallace independently came up with the idea of natural selection and published work on the subject together with Darwin in 1858. This prompted Darwin to publish On the Origin of Species the following year. Wallace made observations as he traveled around the world that provided lots of evidence to support the theory of evolution by natural selection. For example, he is most famous for his work on warning colors on insects which were used by some species to deter predators from eating them. Wallace's research took him to remote places like the Amazon rainforest and the Malay archipelago, where he collected thousands of specimens. His detailed observations of how species varied between different islands helped build the foundation of our modern understanding of speciation. Understanding speciation helps us see how the incredible diversity of life on Earth has developed. It's the process that has created the millions of different species we see today, and it continues to happen all around us. In our next episode, we'll explore the fascinating evidence for evolution from the fossil record. This episode of our series covers the work of Gregor Mendel on genetics, why his ideas weren't popular at the time, and further developments in genetics after him. Let's begin with Gregor Mendel, an Austrian monk whose work in the 1860s laid the foundation for modern genetics. He carried out a series of breeding experiments using pea plants, selecting them because they had clear, contrasting features. What made Mendel's approach special was that he carefully tracked specific traits such as plant height, flower color, and seed shape through multiple generations. Think about it. He literally counted thousands of individual pea plants and precisely recorded their characteristics. Through these painstaking experiments, Mendel observed that traits weren't simply blended together in offspring as was commonly believed at the time. Instead, traits appeared to be passed down as discrete units that remained unchanged from generation to generation. Mendel called these units factors, what we now know as genes. He concluded that each trait was determined by two factors, one inherited from each parent, and that some factors were dominant over others. For example, when he crossed tall pea plants with short ones, the offspring weren't medium height as people expected. Instead, they were all tall, showing that the tall factor was dominant over the short factor. Despite publishing his findings in 1866, Mendel's work remained largely unrecognized during his lifetime. But why was such groundbreaking research ignored? There were several reasons for this scientific oversight. First, Mendel published his work in an obscure scientific journal that wasn't widely read by the international scientific community. Second, his statistical approach to biology was unusual for the time. Most biologists weren't mathematically trained and struggled to understand his numerical analysis. The idea of using maths to study living things was quite revolutionary. Perhaps most importantly, the scientific community simply wasn't ready for Mendel's ideas. His theories contradicted the prevailing belief that inherited traits were blended together, like mixing paint colors. There was also no understanding of the physical basis for his mysterious factors. Scientists couldn't see or touch them. It wasn't until the early 20th century that the significance of his work was finally recognized. The rediscovery of Mendel's research coincided perfectly with new observations about cell biology. In the late 19th century, scientists had developed much better microscopes that allowed them to observe chromosomes during cell division. They could see these thread like structures moving around inside cells, but didn't yet understand what they did. This was like having a key but not knowing what door it unlocked. In the early 20th century, scientists began to notice something remarkable. Chromosomes and Mendel's factors behaved in remarkably similar ways during reproduction. Both seemed to be passed from parent to offspring in predictable patterns. This led to the crucial insight that genes might actually be located on chromosomes. This connection between Mendel's abstract factors and the physical structures inside cells was a major breakthrough in scientific understanding. Scientists now had a mechanism to explain how true traits could be inherited. The chromosomes were like tiny filing cabinets carrying genetic information. But the story doesn't end there. In the mid 20th century, another huge advance came with the discovery of DNA structure by Watson and Crick in 1953. This built on the work of many other scientists, including Rosalind Franklin, whose X-ray crystallography was crucial to understanding DNA's shape. Once the double helix structure of DNA was understood, scientists could begin to work out exactly how genes function at the molecular level. They discovered that genes contain the instructions for making proteins, which carry out most of the important functions in our cells. All of this scientific work by many different scientists over more than a century led to the development of what we now call gene theory. This is our modern understanding of how characteristics are inherited through genes and how they function in our bodies. This story of the development of genetic understanding is a perfect example of how science progresses. Ideas are proposed, tested, sometimes forgotten, then rediscovered and refined as technology and knowledge advance. Scientific understanding rarely comes from a single breakthrough, but develops gradually through the collective work of many scientists over time. Each discovery builds upon previous work, creating an ever growing tower of knowledge. Our next episode will explore the evidence for evolution, including fossils and antibiotic resistance in bacteria. This episode will cover what fossils actually are, the different ways they form, how they show organisms change over time, and why there are gaps in our understanding. So, what exactly are fossils? Fossils are the preserved remains or traces of organisms that lived long ago. They're typically found embedded in rock and can be millions of years old. Fossils are incredibly valuable to scientists because they provide evidence of what ancient creatures looked like and how they've evolved over time. They help us piece together the history of life on Earth. There are three main ways that fossils can form. Let's take a look at each of these processes. The first and most common way fossils form is through gradual replacement of minerals. This happens when parts of an organism that don't easily decay, such as bones, shells, and teeth, are buried in sediment. Over very long periods, these hard parts are gradually replaced by minerals as they decay. The minerals form a rock-like substance that takes the shape of the original body part. Eventually, the surrounding sediments turn to rock, with the fossil clearly visible within it. When someone discovers and carefully excavates the rock, the fossil is revealed. The second way fossils can form is through casts and impressions. Sometimes, an organism may leave an imprint in soft material like mud or clay. Later, material hardens around the imprinted area, leaving behind a cast. Things like footprints, burrows, or plant roots can also create impressions in soft sediments, which are preserved when the material hardens. These trace fossils don't contain any actual parts of the organism, but they provide evidence of its existence and behavior. The third way fossils form is through preservation in environments where decay doesn't occur. In certain special places, organisms can be preserved almost intact. For example, in amber, fossilized tree resin, insects and small creatures can be trapped and preserved with remarkable detail. The resin prevents oxygen and decay microbes from reaching the organism. Similarly, in glaciers, the extreme cold stops decay microbes from working. In peat bogs, the highly acidic conditions prevent decay organisms from breaking down tissue. A remarkable example of this type of preservation was the discovery of woolly mammoths in Siberian permafrost, with some specimens so well preserved that their skin, hair, and even internal organs remained intact after thousands of years. Fossils give us crucial information about how living things have changed as life has developed on Earth over millions of years. By comparing fossils from different time periods, scientists can track how species have evolved. However, despite all we've learned from fossils, there's still a major mystery. How did life begin on Earth? There are various scientific hypotheses about how the first living things appeared, but there's no complete consensus. Some suggest that the first life emerged in primordial oceans or near deep sea vents. Perhaps simple organic molecules came together on early Earth, combined to form more complex organic molecules, and eventually developed into very simple life forms. These hypotheses are difficult to confirm because we lack solid evidence from that time period. Many early forms of life were soft bodied, with tissues that easily decayed. This means the fossil record for the earliest life forms is incomplete. Additionally, many fossils that did form millions of years ago may have been destroyed by geological activity, such as the movement of tectonic plates. The fossil record is a bit like a book with missing pages. Scientists can read the parts that remain and try to piece together the complete story, but there will always be gaps in knowledge. Despite these limitations, the study of fossils has revolutionized our understanding of life's history on Earth. Each new fossil discovery adds another piece to this grand puzzle, helping us better understand the amazing story of life on our planet. In this episode of our series, we'll explore when antibiotic medicine started, how bacteria began outsmarting antibiotics, antibiotic resistance, and finally, how the world is responding to this issue. Let's begin with one of medicine's greatest success stories. The discovery of antibiotics like penicillin was revolutionary. Before antibiotics, a simple cut could kill you if it got infected. Imagine living with that constant fear. Suddenly, bacterial infections that had been death sentences became curable with pills. But here's where things get interesting. Bacteria have developed an incredible survival trick. They can evolve to resist the very drugs designed to destroy them. How on earth does this happen? Like all living things, bacteria sometimes get random mutations in their DNA. Think of it as nature rolling the dice. Most mutations do nothing, but occasionally one hits the jackpot. Some lucky bacteria might develop the ability to break down antibiotics like they're digesting food. Others evolve tiny pumps that literally push the antibiotic out of their cells before it can do damage. This is how bacteria develop resistance against medicines. And once they have these traits, they pass them on to their offspring. How wonderful. When you take antibiotics, the non resistant bacteria die off quickly, but those resistant ones survive, thrive, and have the whole environment to themselves. With all their competition dead, resistant bacteria multiply like crazy. They can produce millions of copies of themselves in just hours. We're talking about new generations every 20 minutes. Plus, they can share genetic information with each other in the form of plasmids. This means resistance can spread through bacterial populations at lightning speed. One resistant bacterium can quickly become millions, all carrying the same resistance genes. For people infected with these superbugs, standard treatments become less helpful. Take MRSA, for example. This notorious superbug can be incredibly difficult to treat. If MRSA enters your bloodstream, it can be fatal. For decades, we've been winning the war against bacterial infections. Death rates from diseases like pneumonia have plummeted thanks to antibiotics. But now we're losing ground, and it's partly our own fault. Doctors sometimes prescribe antibiotics for minor illnesses or viral infections like colds and flu. Here's the problem: antibiotics can't kill viruses. It's like trying to use a hammer on a screw. Additionally, patients make things worse by not finishing prescriptions. They feel better after a few days and stop taking the pills, but this leaves the strongest bacteria alive. Every time we use antibiotics incorrectly, we're essentially training bacteria to become resistant. We're creating the perfect conditions for superbugs to evolve. Remember, antibiotics don't create resistance directly. They simply kill off the weak bacteria, leaving the playground clear for resistant ones to take over. That's why finishing your full course of antibiotics is crucial. It ensures all bacteria are destroyed, not just the weak ones. The problem extends beyond hospitals and clinics. On farms, antibiotics are sometimes given to healthy animals to make them grow faster and prevent illness. This creates a breeding ground for resistant bacteria. These superbugs can then jump to humans through meat production and consumption. Some countries have started restricting agricultural antibiotic use, recognizing the danger. But globally, we still use massive amounts of antibiotics in farming. Pharmaceutical companies are racing to develop new antibiotics that can defeat resistant strains. But here's the catch: creating new drugs is painfully slow and expensive. This is why prevention is vital. Using antibiotics only when truly necessary, completing full treatments, and limiting agricultural use can all slow resistance development. What we're witnessing is evolution in action, natural selection at hyperspeed. Bacteria are adapting to survive in a world filled with antibiotics. Just as Darwin predicted, organisms would adapt to environmental changes. Next time, we'll explore how scientists organize and classify the incredible diversity of life on Earth. In this episode of our series, we'll look at why classification is important, the Linnaean system, how molecular biology led to new systems, and evolutionary trees. Let's explore the principles behind biological classification. Classification is fundamental to how humans process information, from organizing libraries to structuring databases. In biology, this organizational instinct becomes a vital scientific tool. By grouping the sheer number of organisms on Earth based on shared characteristics, scientists can identify patterns and understand relationships between different forms of life. Without classification, biological research would lack the structure needed for meaningful analysis. The modern classification system originated with Carl Linnaeus, an 18th century Swedish scientist. He developed the Linnaean system, which arranges organisms into nested groups, each level more specific than the last. This hierarchical structure creates a logical framework for understanding biological diversity. The classification hierarchy consists of 7 main levels: kingdom, phylum, class, order, family, genus, and species. Each level represents a different degree of relatedness between organisms. This system functions like a postal address, beginning with broad categories and progressively narrowing down. Just as an address moves from country to specific street number, classification moves from kingdom to individual species. For over 2 centuries, this framework provided the foundation for biological classification. Traditionally, scientists recognized 5 kingdoms: animals, plants, fungi, protists, and bacteria. This division was based primarily on visible characteristics and basic cellular structure. However, advances in microscopy and molecular biology revealed that traditional classification based on physical appearance didn't accurately reflect evolutionary relationships. Scientists discovered that organisms appearing similar externally could be very different at the molecular level. In 1990, Carl Woese proposed a revolutionary free domain system based on genetic analysis, which transformed our understanding of life's most basic organization. Through RNA sequence analysis made possible by new technology, Woese identified 3 fundamental divisions of life that supersede the kingdom level. The 3 domains are archaea, bacteria, and eukaryota. These represent the earliest evolutionary branches in the tree of life. Archaea are single-celled prokaryotes often found in extreme environments, such as thermal vents and salt lakes. Despite superficial similarities to bacteria, they possess unique biochemical characteristics. These organisms demonstrate remarkable adaptations to conditions lethal to most other life forms. The bacteria domain encompasses true bacteria, including familiar species like E. coli and Staphylococcus. Though prokaryotic like archaea, bacteria have distinct molecular and biochemical properties. Eukaryotes include all organisms with nucleated cells and membrane-bound organelles. This domain contains the kingdoms of fungi, plants, animals, and protists. This classification reveals surprising relationships. Humans share more fundamental characteristics with fungi than bacteria share with archaea. Such insights demonstrate the value of molecular classification. Within these 3 domains, the traditional Linnaean hierarchy remains useful. The system continues to provide detailed classification below the domain level. Scientific naming follows strict international conventions to ensure clarity. The binomial nomenclature system assigns each species a 2-part Latin name. The first part indicates the genus, while the second identifies the specific species. For example, humans are designated Homo sapiens, Homo denoting our genus, and sapiens our species. This systematic approach eliminates vagueness in scientific communication. Binomial nomenclature provides a universal language for scientists worldwide. Regardless of native language or location, researchers use identical names for the same organisms. Evolutionary trees visually represent relationships between species based on common ancestry. These tools help scientists understand how different organisms evolved from shared ancestors. Branch points on these trees indicate common ancestors, with closer branches suggesting more recent divergence. The structure reveals evolutionary relationships that may not be apparent from physical characteristics alone. Scientists construct these trees using multiple data sources, including DNA sequences, protein structures, and fossil evidence. Modern computational methods allow analysis of vast amounts of genetic data. This comprehensive approach provides insights into evolutionary history spanning billions of years. By integrating various evidence types, scientists can trace relationships from microorganisms to complex multicellular life. Next time, we'll examine biodiversity and explore why maintaining species variety is critical for ecosystem stability.